a Google Drive in the chat, and this Google Drive is going to contain the data set that we're going to be using for our workshop today, as well as the slides and link to the Kaggle so you guys can follow along. So as I previously mentioned, my name is Tarun Suresh, and I'm from Mission San Jose High School, and I'll be introducing you guys to computational neuroscience. Data of inflammatory biomarkers. So let's jump right in. So let me give you guys a brief overview of what all we're going to be covering today. So first, I'll be teaching you guys about like the basics of Alzheimer's disease, what it is, and why it's so important to build diagnostic models for Alzheimer's.
Hi, Taryn. Sorry to interrupt. We're getting a bit of a signal issue, so I'm going to, if you wouldn't mind, I'm going to turn off your camera just to save some bandwidth. Perfect. Thank you very much.
Okay, going back to it. So I'll be giving you guys an overview of the biomarker data set that we're going to be using to make our predictions. Following that, I'm going to be introducing you guys to Kaggle and how to code in Kaggle and develop a notebook in Kaggle. And then we're going to start loading our data, and we're going to perform some basic data wrangling. Following that, I'm going to introduce you guys to supervised machine learning in Python. Then I'm going to walk you through a specific type of supervised machine learning algorithm called boosting algorithms and extreme gradient boosting. And then we're going to develop our own extreme gradient boosting, XGBoost model. Following that, we're going to find the most optimal hyperparameters through a specific hyperparameter tuning technique called evolutionary computation. Following that, we're going to optimize our model using the optimal model hyperparameters that we found from our evolutionary computation. Lastly, we're going to explain our model through Shapley additive explanations, and we're going to interpret the most important features of our model that contribute to predicting whether a patient has Alzheimer's or not. So what is Alzheimer's disease? It is a progressive brain disease that destroys memory, thinking, and behavior skills. It impacts 5.5 million Americans, and it costs the US $277 billion each year. So currently, there's no magic bullet for Alzheimer's. There's no definitive treatment or cure. But an earlier diagnosis can provide a better chance of benefiting from the treatments available. And that's why we're going to be developing an Alzheimer's diagnosis model using machine learning today. In order to do so, we're going to use a specific data set. And this data set is an inflammatory biomarker and personal data from Alzheimer's disease and healthy control participants. It's adapted from a data set published by Beska et al. On the open access Zenodo repository. And so our columns include the class, whether a person has Alzheimer's or not, the participant ID from the study, age, gender, and all these 10 biomarkers: interleukin 1, interleukin 6, tumor necrosis factor, prostaglandin J2, prostaglandin E2, brain-derived neurotrophic factor, nerve growth factor, MDA, nitrites, and glutathione. So as I said, our data set contains the subject class. And so we have two classes. A total of 54 participants were in the study, and we have 35 Alzheimer's participants and 19 healthy control participants. So we have more Alzheimer's participants in our study, as you can see in the figure. We also have in our data set the ID number from the study, from the study by Beska et al. 2020. And this is not relevant for our modeling. So when we do our data wrangling, we're going to drop this column. And our next column contains the ages of the subjects. And the ages range from 56 to 89. And if you look at the mean, you can see that most of the mean age of our subjects is 77, so quite old. We also have the gender of the subjects of the data set. So we have 25 female, and they belong to gender class 1, and 29 male belong to gender class 2. Additionally, we have concentrations of brain-derived neurotrophic factor. And I'm just going to show you one. I'm just going to show you one of these. I just picked brain-derived neurotrophic factor. And so a brain-derived neurotrophic factor is a member of the neurotrophin family. It functions in the brain to promote growth, maturation, and maintenance of nerve cells. So we're going to be investigating how this biomarker and many other biomarkers and the personal data contributes to predicting whether a patient has Alzheimer's or not. So now let's get started with coding. Please go to this website, www.kaggle.com. You can also find this presentation and the Kaggle link in the Google Drive that I shared in the chat. So everyone, please go to this Kaggle link. After that, you need to either register or sign in, and you can register with your email to make it easier. So I'll give you guys like a minute or two to go to Kaggle and okay. Okay, hopefully left-hand side, you're going to see this option that says code. You're going to click on that, and then you're going to click on a new notebook. That's going to create our new notebook that we're going to use for our coding. I've also attached a link to the complete Kaggle notebook that I made for this prediction, and it's available as a Google Doc. It's a link in a Google Doc, or it's in this slideshow presentation if you want to access it. Okay, I'm going to move on now. If you guys get stuck, you guys can ask Viren for any help, or you can also follow the presentation that I have put in a drive or the Kaggle link. Both of them have the complete set of instructions. get into the drive folder, there's going to be a CSV file. So could you guys download that CSV file to your computer? Because we're going to start uploading our data into Kaggle, and we need to download a CSV file to do that. Okay, once the CSV file downloads in the Kaggle notebook, you're going to click on File, and then you're going to click on Add or Upload Data. Then you're going to select Upload, and then you're going to select Browse Files from your computer. And then in your computer, you're going to select the Alzheimer's CSV that you downloaded. And then for convenience, you can name your data as Alzheimer's or however so you choose. And then you're going to click on Create. Okay, once you upload your data into Kaggle, so I'm gonna show you guys two basic functions of your Kaggle notebook. One is the scissors icon. It's gonna be on the top. And just click on the scissors icon to delete that. And then you're gonna click on the plus icon to add a new cell. And then you're gonna enter the following code into the new cell. And this is gonna create a path so that when we load a CSV using pd.read CSV, we're gonna have a specific path, and these lines of code is gonna print out that path to use. Lastly, you're gonna click the play button to run the code in this cell. Okay, hopefully you guys were able to load the data. Now let's transform our data into a more usable form through the process of data wrangling. This will involve removing certain rows with null values and dropping columns that are not needed. We're going to drop the ID column. And so working with data sets in Python is going to involve the package library called pandas. So first in Kaggle, you're going to click on the plus icon to add a new cell. And then in that new cell, we're going to import two libraries, NumPy, numerical Python, and pandas. And so we're going to create a variable called df, which will store our data frame. And we're going to say pd.read CSV. So we're going to read the CSV file that we uploaded to Kaggle. Following that, we're going to drop our ID column using df.drop. x is equal to 0, and any and rows that contain any null values will be dropped. And then we're going to output the head of the data. So it's going to show like the first few rows and columns of our modified data set. And then in order to run the code, always remember to click play so you can run the code in that particular cell. We can run all the cells. Okay, hopefully you guys were able to run that code. I'm going to be moving on to splitting our data. So as I previously mentioned, we're going to be working on machine learning classification. And so for machine learning classification, we have to split our data into features, which are the inputs, and labels, which are the outputs. And so our features will be in the X data frame, and our labels will be in the Y data frame. So our features are going to be the age, gender, and all 10 of the biomarkers in those 10 specific columns. And then our label or output is going to be the class. So either a person is healthy or has Alzheimer's. So in order to split the data, we're going to enter in a new cell. So add a new cell, and then we'll call X, capital X, is equal to df, our data frame, dot drop, the subject column. So we're going to include all the columns except for the column with the subject, because the subject column contains the class, either a person is healthy or has Alzheimer's. And then so our Y is just going to be the subject column. So zeros for healthy or ones for Alzheimer's. And then once again, click the play button to run the code in the cell. Okay, now let's move on. We're going to split our data even further. We're going to split our data into train and test. Train data is the data that the machine learning model is going to use to learn from. And then we're going to evaluate our machine learning model using our test data. So 20% of our data is going to go toward testing the model, while the remaining 80% will be used to train the model. So in a new cell, we're going to import train test split from scikit-learn.model selection. We're going to split our X and Y data frames even further into X train, X test, Y train, and Y test. And so 20% of our data, as I previously mentioned, will go towards testing. We're going to also have a random state parameter. And so we pass an integer, and this will split our data in a certain way that it's reproducible. And then you're going to click the play button to run the code in the cell. Okay, moving on. So let me briefly digress to what is machine learning, because I've been talking about it a bit and I didn't give a formal introduction to what machine learning is. So machine learning involves computer algorithms that learn from data. So these computer algorithms are going to make decisions and predictions without explicitly being programmed to do so. One major category of machine learning is supervised learning. And this involves mathematical modeling out of data that contains structured inputs and outputs. One type of supervised learning is classification. And this involves assigning a label to an observation based on its features. For example, we can assign an email as a spam or not spam based on certain words in the email. For instance, if the email says like click on this link to maybe win some prize, maybe that those certain words uh, make it more likely to be a spam email than a not spam email, or diagnosing a patient based on certain characteristics, which is what we're doing right now in this particular workshop. Maybe one biomarker is significantly higher, or certain biomarkers are significantly higher in Alzheimer's, and that's why a patient is diagnosed with Alzheimer's. And so one type of machine learning algorithms, class of machine learning algorithms for supervised learning is boosting. And so a boosting basically is a type of ensemble machine learning algorithm. And so it ensembles weak learners to yield an even stronger learner. And so one type of boosting algorithm that's optimized is, and it's really popular, it has it's a Kaggle winning machine learning algorithm and usually has high accuracy and model performance, is extreme gradient boosting, XGBoost. And so it optimizes our gradient boosting through parallel processing, tree pruning, handling missing values, and regularization to reduce our overfitting and bias and improve our accuracy and model performance in general. So now we're going to implement our XGBoost. So in Kaggle, click on the plus icon to add a new cell. In the new cell, add the following code. We're going to be importing XGBoost as XGB. We're going to be also importing two metrics, accuracy score and classification report. Accuracy score is the accuracy of the predictions made by the model. Classification report gives like a table like thing of like many different metrics such as accuracy, F1 score, precision, and recall for both classes. So we're going to see like this overview of like so many different metrics. So it's really helpful to evaluate our model. And so to create a model, we're going to create a variable called model. And so model is equal to xgb.xgb classifier. And we're going to fit our training data, so our X train and Y train. Then we're going to print our accuracy. We're going to print our accuracy score, and we're also going to print our classification report. So click the play button to run the code in the cell after copying all this code down. And if you ever get lost, you can check out the Kaggle link, or you can consult this presentation that I've linked in the drive, or you can ask Viren for any help. Okay, now let's look at the results of our XGBoost model. So we're going to see over here that our accuracy was about 55%. about 0.55, and this is a low accuracy score. So that means our model is not very accurate at predicting whether a patient has Alzheimer's or not. Our predictions are almost close to chance. You can also see that our recall for healthy patients is very low. It's only 0.2. So in order to resolve this, we're going to do something called hyperparameter tuning. And so this is going to increase our model performance by finding the most optimal parameters for the machine learning model to get the best accuracy possible. And so one way to do so is through a hyperparameter tuning technique called evolutionary computation. And so evolutionary computation finds the best parameters using principles of evolution and natural selection. And you can see an image of our evolutionary computation over here, just like a general workflow of what happens. So now we're going to implement our evolutionary computation. And so we're going to be, as I previously said, we're going to be optimizing our model for the highest accuracy possible. So we're going to copy the code from cells 5 and 6 in the Kaggle link, since it's pretty long to paste over here in the slides. I've linked the Kaggle for reference. You guys can copy down the code. And you're going to click the play button to run the code in the cell for each cell. And so the second cell might take a while. So cell 6 might take a while to run because it's going through all the parameters and all the generations, all 10 generations that I'll be going through. And so in that time, in the meantime, I'm going to be talking a little bit about what this evolutionary computation is. So evolutionary computation, we're going to use something called a genetic algorithm. And this is a hyperparameter tuning technique that takes inspiration from Charles Darwin's theory of natural selection. It can be used to optimize the parameters of a machine learning model to improve model performance using concepts in biology such as mutation, crossover, and selection. Our genetic algorithms is going to start with an initial population of parents. And so each parent is going to have certain traits, which are the parameters of our XGBoost model. And so we have 7 parameters of the XGBoost model. We have we have 7 parameters of our XGBoost model. And then so the first thing that happens is we first run this, and then we run it, and then it assesses based on our initial machine learning models, like the initial parameters, how accurate these are. And so this will generate a particular fitness score or accuracy score. And so this fitness score can be any model performance, such as accuracy, F1 score. In this case, we're going to use accuracy score for our genetic algorithm. And so based on the fitness scores in this initial 0 generation, the top performing parents are going to be selected. And this is similar to like survival of the fittest in Darwin's theory of evolution and natural selection. So the parents with the certain parameters, like the best performing parameters, are selected. So they survive, and they can now mate to produce offspring. And this mating process is going to involve 2 things: crossover and recombination. It's also going to involve mutation. So in crossover, the machine learning parameters from 2 different parents, survived parents that mate, they're going to be combined to produce offspring. And each offspring is going to inherit some parameters from each parent. And then also our genetic algorithm is going to introduce some mutation. And so some of the parameters are randomly going to be altered to maintain our genetic diversity. And it's going to enable our genetic algorithm to output parameters that yield the highest fitness score possible. And so this entire process is going to repeat 10 times for each generation. And then it's going to continue until all these generations run and the best fitness score with the optimal hyperparameters will be yielded. So we're going to have 64 parents, 32 of which will be mating to produce offspring. We're going to use all 7 parameters of the XGBoost model, and we're going to have 10 generations of individuals. So after running the code, we're going to see that our best fitness score is about 82%. And this is a significant jump because if you recall from before, our fitness score was about 55%. And then we're going to see that our best parameters, the 7 parameters we used, learning rate, N estimators, max depth, min child weight, gamma, subsample, and co-sample by tree. We're going to see all the optimal parameters for this that yielded this high accuracy score. Okay, now let's optimize our XGBoost model. So the first thing you guys are going to do is in our Kaggle notebook, we're going to click on the plus icon to add a new cell. In that new cell, you're going to add the following code. You're going to develop a new model with the parameters from before, like the learning rate, estimators, max depth, and such. And then we're going to fit our training data. And then we're going to print our accuracy score and classification report, as we did before with our original model. And then note that the parameter values that you got from the previous step, from the previous genetic algorithm, may differ. So use the ones that were yielded by your genetic algorithm when you ran the code. So I'll give you guys a couple minutes to run that. Okay, let's move on. So now here we can see our results from running this optimized XGBoost model. We can see that the accuracy has improved significantly, as mentioned previously, from 55% all the way up to approximately 82%. We're also going to see that our precision and recall scores for the 0 and 1 classes, so for our healthy and Alzheimer classes, are 1. So it's like perfect precision and recall scores. So now what we're going to do is we're going to perform a visualization of feature importance. And so feature importance is going to be used to explain visually how each feature contributes to the predictions made by the model. And so we're going to use something called shapely additive explanations. And this is a game theory approach to feature importance. So in Kaggle, click on the plus icon to add a new cell. In a new cell, you're going to add the following code. So first, since this SHAP library is not in the Kaggle environment before, you're going to have to install it using this pip install command. And then after that, once that installs, you're going to import SHAP. And then you're going to create a variable called explainer. And it's going to be explaining the features of the previous model, the CLF variable, or previous model. And then we're going to create a horizontal bar graph of our shapely values. And you're going to click play to run the code in the cell. Okay, let's move on to our results of our feature importance using shapely additive explanations. And so what these machine learning models do, they can actually give us they can actually show us which features are most important to predicting a certain like our labels at the end. We can use this for future research. Like we can say that certain features are extremely important to predicting Alzheimer's over other features. For instance, our prostaglandin J2 is our most important feature for this XG boost model to predict Alzheimer's. Following that, we have tumor necrosis factor, and then age, and then nerve growth factor, then prostaglandin E2, and then we have brain-derived neurotrophic factor, and then we have our two inflammatory interleukins, IL-6 and IL-1. Then we have nitrites, and then we have three other features that are not really important. Then they have sum. The total of those three features is going to be 0.13. That's their shapely value. So let's interpret these feature importance results and see how they align with other research studies. And so prostaglandin J2 was our most important feature for predicting Alzheimer's for this XG boost model. And so prostaglandin J2 is a neurotoxic protein, and it's going to induce processes relevant to Alzheimer's, such as promoting the formation of amyloid beta plaques. And so amyloid beta plaques are like hallmarks of Alzheimer's. They're known to be present in people with Alzheimer's. And then we also have our second most important feature is tumor necrosis factor. And this aligns with previous research that shows that tumor necrosis factor signaling worsens the amyloid beta plaques and phosphorylated tau proteins, which are hallmarks of Alzheimer's. And so we can see that this protein is also really important for whether a person has Alzheimer's or not, because it's involved in all these inflammatory and neurodegenerative pathways that are pretty important for Alzheimer's. Our third most important feature is age. And so age is considered the greatest risk factor for developing Alzheimer's. And so our symptoms and risk worsen as a person ages for Alzheimer's, since it's a neurodegenerative disease and more neurons are destroyed over time as a disease progresses. And so I have references of the PESCA data set and any questions you guys may have. Remember, I have provided you with a framework to develop a simple XGBoost model. You can always improve on this model using feature engineering, such as and dimensionality reduction, such as principal component analysis. You can try out different algorithms, machine learning algorithms, such as logistic regression, decision trees, random forests, support vector machines. You can even try out deep learning or super learning and try to improve model performance. So any questions you guys may have, please feel free to share them. That's pretty much it for my presentation.